All right, folks, it's now 12 p.m., so we're going to go ahead and get started. Thank you so much for joining us today. This is the first of 3 webinars we'll be hosting this fall. If you haven't already, you can register for the November and December webinars at ntbg.org/events, and I'll be dropping that link into the chat shortly as well. This webinar is being recorded. We'll send the recording out to you in the next few days. You're welcome to ask questions in the chat box throughout the presentation, and we'll also hold a general Q&A at the end. Try to get to all of your questions, but if we don't, please feel free to follow up with me via email, which I'll drop into the chat as well. We're hosting today's session from our Botanical Research Center here on Kauai. I have with me Dr. Nina Ronstedt, our Director of Science and Conservation, who will tell you a bit about today's session. Go ahead, Nina.
Thank you, Amanda, and welcome to all of you. We're so excited to have you joining us today. With this new series of webinars, we want to invite you behind the scenes to meet some of the people who are leading NTBG science and conservation work. As you may already know, Hawaii's flora is really unique, and most of the species are found nowhere else on Earth. However, our native plants and animals are also threatened. In fact, more than 130 plant species have become extinct in the last 175 years. But luckily, there's a lot more awareness of this challenge today, and we are working with many partners across the state to help stop extinctions and restore our native forests. And this series of webinars will zoom in on some of the different aspects of this work. One of the most important steps in saving plants is to collect and safely store seeds so they can be used to be planted back out into nature. And today, we'll be visiting NTBG's seed bank and lab manager, Dustin, who's doing a really amazing job trying to take care of more than 16 million seeds from over 500 native Hawaiian plant species, some of which only exist in these collections. So Dustin is going to start with a brief presentation on some of the research that's being done in our laboratory to make the seed banking of rare and endangered plants possible, before giving you a live tour of the facility. And lastly, he will tell us more about the efforts to save Hawaii's sacred Ohia trees. So welcome again.
All right, can you see my screen? Yes, okay, great. All right, so that was a great introduction. Thank you, Nina, thanks, Amanda, and let's just get right into it. Hawaii is known as a hotspot of biodiversity. So we have about as many naturalized plant taxa as there are native plant taxa. But of those native taxa, about 90% of those are endemic, which means that 90% of these species occur nowhere else on Earth. Looking at the Endangered Species Act listed species, Hawaii has about 0.1, less than 0.1% of the landmass of the rest of the United States, but over half of all the ESA listed endangered species occur in Hawaii. And looking at Hawaii specifically, we've recently assessed all of our 255 Kauai Island endemic vascular plant taxa using the IUCN Red List of threatened species. And we found that 100% of our Kauai endemic vascular plant taxa fall into a threatened category, with 10% of those already extinct or extinct in the wild. The global strategy for plant conservation is a program of the United Nations Environmental Program and provides a framework for plant conservation. Target 8 of this strategy states that at least 75% of threatened plant species should be preserved ex situ. This is also the target that you can think of as banking on nature or a genetic safety net. And so seed banking is the most efficient and cost-effective way to have this kind of collection. However, there are certain things to think about when trying to conserve wild species versus cultivated species. So here we have a picture of a nice Hawaiian forest on the left and a generic photo of an agricultural field somewhere. And these differences are summed up pretty nicely in this table. But if you are a farmer, you're looking for consistency. You want to plant your field all at the same time and have the plants grow at the same time so you can harvest at the same time and have very standardized shapes, sizes, flavors, textures, all those things. Whereas when we're conserving wild species, what we're actually looking for is that genetic diversity. So we want a diversity of different characteristics. We don't want everything to be super standardized. Looking at the NTBG seed bank and laboratory, we have just over 16 million seeds from about 4,700 accessions. The vast majority of those are Hawaiian endemics, but we also do have some Polynesian introduced plants. And we don't just store seeds, we also store fern spores and pollen as well, although the overwhelming majority of our collection is seed. In this newish book on fern conservation, we were listed as one of only 8 active fern spore banks in the world. Certainly there are more, but we're listed as one of them. It's great. And then here we see one of our students or interns doing some pollen conservation in the lab. And this is our lab here. So we'll get to see it in more detail in the next session. And so one thing that you need to know is that seed survival curves inform seed bank management. So this is what a typical seed survival curve might look like. You'll see there's a period of high viability somewhere in the beginning, and then there's a threshold in which viability rapidly declines. And so it's kind of my job to understand what that looks like. In order to do that, we routinely test viability to assess longevity. Our very first seed accession is from 1989, and we've been storing seeds every year since. So we have the oldest known collection in Hawaii, and we're able to kind of reach back in time and pull out some of these data. So we, along with some other seed banks in the state, recently assessed 295 species in the Hawaiian flora. And there's this idea of a recollection interval. That's the time where the viability of a collection or an accession falls to some predetermined threshold. You withdraw those seeds, propagate them, and then you go back out to that plant or that population, if they still exist, and store those, thereby refreshing the seed bank collection. So we found that at the time of the most recent test, 195 of those species had not yet reached their recollection interval, which means that 100 species had, and 45 species had a recollection interval of less than 5 years. And here at the NTBG seed bank, because we're doing this routine viability testing, some of our 30-year-old seeds have recently come up for testing. And so far, 7 endangered species of seeds have already germinated. So that gives us hope that we were doing something right in the earliest days of our seed conservation efforts, and that these species are available for restoration purposes. One thing that we do to try to extend the longevity or preserve seeds is we can kind of cool and dry them. And so this chart here shows the phase state changes of water and what all inside of the seed, and what I'll show is what happens with the orthodox seeded species, which I'll explain in a second. So here we have a happy little seed, and we harvest it in the ambient environment off the mother plant. So high temperature, which we have on the Y, and high moisture, which we have on the X. And if we were to take this seed and just stick it into the freezer, then we would be creating ice crystals. And so those ice crystals are lethal to the cells of the seeds. And so we don't do that. So what we do is we harvest our seeds, and first we desiccate or dry the seeds. Then, then we cool them down below sub-freezing temperatures, thereby achieving this amorphous glass or a viscoelastic. And that's where the water molecules just kind of come together in place, forming this amorphous glass. And that's good news if you're trying to preserve seeds. However, not all seeds behave the same way when you try to apply these things to them. So there's orthodox seeded species, that's what we just talked about. And that's the vast majority of kind of plant species everywhere, especially in temperate areas. And then there are recalcitrant seeded species. These are species whose seeds do not tolerate any kind of desiccation or drying, and so they therefore don't survive any kind of sub-freezing temperatures. But then there's also this intermediate category, which can manifest in one of 3 different ways. There's intermediate, desiccation intermediate, which is just like what it sounds like. The seeds survive more desiccation than a recalcitrant treated species, but less than an orthodox. There's intermediate, short-lived, regardless of how they're dried or cooled. That's like some alpine species. And then there's intermediate, freeze sensitive. So these are seeds that are apparently desiccation tolerant, just like an orthodox seeded species, but they decline faster at sub-freezing temperatures compared to cool temperatures. And it takes some time in order to determine this divergence, time like 2 to 5 years. So this doesn't happen overnight. And so we looked at this in the Hawaiian flora, some of the flowers of, in the Hawaiian flora. And we found that this kind of peculiar seed storage behavior was happening in a few different families. The one here is shown is the bellflower family, or Campanulaceae. And if you're below the line, you're likely freeze sensitive. And if you're above the line, you're likely not. So we found this in all genera of the Campanulaceae family with perhaps maybe not this Stromatolobelia, which is somewhat interesting. We also found this in the Cyperaceae or sedge family, in the Rubiaceae or coffee family, and in the Urticaceae or nettle family. And so it turns out that Hawaii has the highest proportion of any flora ever assessed exhibiting this intermediate freeze-sensitive seed storage behavior. So Hawaii can really be a model for other island, tropical, and subtropical systems in seed conservation. We then took all of these data and we made them available online at the website seen here, hosted by Laukahi, the Hawaii Plant Conservation Network. And so this is a great example of many different institutions working across the state to help implement the global strategy and the Hawaii strategy for plant conservation. So if you're wondering why this might be with these intermediate freeze-sensitive seed storage behavior, we have this method called differential scanning calorimetry. So this is where we cool the seeds down to about -150 and then warm them up to about 50 and then measure the energy output of those. So in this case, it's heat flow. And if we look at that warming scan, we'll see kind of a spike around 0. And we think that is the water going from a solid to a liquid, so a phase state change. But then we also have this other one around -20 or -18, and that is conventional seed storage temperatures. That's our seed storage temperature here for orthodox seeds and kind of across all seed banks everywhere. And so we think what's happening is that these are oily seeds and that this is a lipid phase state change. So these are the lipids going from a solid to a liquid. And because this is happening at our storage temperature, that we're getting this mixing effect. So you get this freezing, thawing, freezing, thawing, freezing, thawing cycles. And then over time like 2 to 5 years, that degrades the cells of the seeds. But this is all unpublished. That was work done at the National Seed Lab in Fort Collins by Chris Walters and her team. And so I look forward to following up on some of that and maybe doing some of this work at Kew, looking at this lipid thermal fingerprinting and also lipid composition as well. And so it's been proposed that herbaria, which are repositories for pressed plant specimens, can be an overlooked or an underutilized resource for harboring extinct species of seeds. Herbaria are managed for preservation of dead plants, not for living plants, as compared to a seed bank, for example. But in this one paper shown here, the authors were able to resurrect an extinct Belgium species after up to 144 years. And so they sowed a few thousand seeds and 8 total seeds germinated. And so they liken this kind of a thing to finding needles in a haystack. But if you don't look through the haystacks, you're not going to find the needles. Okay, the next thing you need to know is about the Plant Extinction Prevention Program, or PEP. This is a program that manages species with fewer than 50 individuals remaining in the wild. And there's actually 257 of those in Hawaii, 81 of which occur on this island, Kauai. Using this idea of looking through the haystack of an herbarium, we had a student intern who generated much of this data shown here. And we looked through our herbarium of the National Tropical Botanical Garden, PTBG, through all of these PEP species. So these 81 species that occur on Kauai. And we ended up looking at about 1,200 specimens, or about 86% of the target taxa. And it turned out that only 138 specimens contain seeds, or 11%. And so there's many reasons why that might be. But we're still able to harvest over 2,000 seeds from those specimens. And those represented about 47% of the target taxa. And it turned out that we did have at least one germination event. That was in the Shidi Kauaiensis from 2017. That was transferred down to our nursery for further propagation. And then we also had these two other embryos present themselves. So a seed is a baby plant in a box with its lunch. The baby plant is the embryo, the box is the seed coat, and the lunch is the endosperm. So in this case, the embryos kind of hopped out. And so that could have been a growth or a living event. In any case, these were also transferred down to the nursery for further propagation. Yes, so moving on, Hawaii is vulnerable to increased salinity from a number of sources. So one, as sea levels rise, salinity at the coast and these coastal dune sites will increase. Also, as we get more frequent storms, we get more frequent storm surge, which will also increase salinity as we're predicted. And also predicted we'll have increased drought, which will also increase salinity. And so we don't know what the salinity tolerance is at our coastal systems, especially at the early life stage of seed germination. So we looked at that in these 20 species. Most of them are common native, they're all native coastal, they're all coastal species. Most of them are native. A couple of them are introduced, and some of them are rare natives. We found that most species could germinate in high salinity, and we used undiluted, unfiltered seawater at different concentrations. And so, yes, most species could germinate in high salinity. We also looked at the timing of germination and found that for most species, germination slowed down with increasing salinity. But we also wanted to test this idea of resilience. So we tested recovery to salinity. So this is where we kind of rinse the seeds in fresh water, all the remaining seeds after some period of time. And so those same seeds in fresh water. And we found that many species can actually recover from at least some salinity. But of course, some species cannot recover at all. Interestingly, our endangered species, Hispania tomentosa, had the highest germination in saline conditions. So that's hope for that species. But at the end of the day, in order to answer all of these questions, we need to get the seeds to germinate. Also, when we pull these seeds out of storage, we have to be able to propagate them by seed in order for them to be useful in restoration and recovery projects. And so we also look at dormancy and germination. So on the left pane here, we have the princess in the pea. And so she needs just the right conditions in order for her to be comfortable. And a seed is much the same. They have to have just the right kind of germination conditions, or they're not going to germinate. For example, seeds in a pack in the shelf on a nursery are probably not going to germinate. And so we call that quiescent. However, if seeds do have all the right kind of germination conditions and they still don't germinate, then we call that dormant. So that's Sleeping Beauty here in the middle. And of course, on the right, we have Jack and the Beanstalk, because once they do have their optimal germination conditions and they're no longer dormant, then we can get nice sprouts, just like the beanstalk here. And so it turns out that there's 5 different classes of dormancy and non-dormancy. And so we've looked at that in some of our Hawaiian species. And so, for example, in these 2 Kauai endemic species of hibiscus, we found that the seeds are non-dormant. So that's like the princess in the pea. And then in this Dianella sandwichensis, we found that the seeds have morphological dormancy. So that's like Sleeping Beauty. We also found an overwhelming majority of tropical montane species in Hawaii have physiological dormancy. Again, that's like Sleeping Beauty. And then in these Hawaiian lobelias, or the Campanulaceae family, the same family I talked about earlier, this is the largest family in the Hawaiian flora. It's also the family with the most extinctions and the most endangered species. We found both morphological and morphophysiological in at least 5 of the 6 genera in this family. So again, we have Sleeping Beauty, but one thing that we haven't assessed is the genus Brighamia, which has become kind of a poster child for plant conservation in Hawaii and elsewhere. And so we're looking at this now, and we found that the embryos do grow inside the seeds prior to germination, and that the seeds will germinate over a broad range of temperatures kind of initially. And so these seeds have morphological dormancy. And so so these kinds of seeds with these Sleeping Beauty might need extra considerations when propagating by seed. And so with that, I think we'll end here and we'll take questions if we have time. Yes?
Yeah, if anybody had any questions for Dustin, feel free to drop them into the chat box. Thanks, Dustin, for that awesome presentation about your research that you're doing here. And let's see, don't see anything coming through yet. So let's go ahead and transition into our tour of the seed lab, and let's see if anything comes in, and we'll do another little question check after that.
Great. I'll meet you all in the seed lab.
Bye, Dustin. We'll see you soon. So I'm going to go ahead and drop those links I mentioned into the chat box for you. So you can go to ntbg.org/events to register for our November and December webinars. And that's my email if you all had any questions and you wanted to follow up with us afterwards. All right, looks like Dustin and Nina are in the seed lab, so I'll let them go ahead and take over.
Okay, yes, Amanda, can you hear me? We're good? All right, awesome, okay. Well, welcome to the seed bank and laboratory. It's in our mission to save plants, and one of the ways you can do that is by having an off-site or ex-situ conservation collection. And it turns out that the most efficient way to house that kind of a collection is to store them as seeds in a seed bank. And so that's what we have here. And when I say most efficient, you can name your metric, whether it's time, dollars, human resources, water, electricity, storage space, most efficient to store them as seeds in a seed bank. And so if you think of biodiversity as a measure of a number of species for some unit area, then we're standing. Oh, well, that's okay. Oh, so I'm small. Oh, I was not there, but they can hear me. Oh, so I should stop sharing. Uh-huh.
Thanks, Dustin. We'll see you again in just a sec.
Okay. Yeah, so you can think of biodiversity as a number of species per unit area, then we're standing in one of the highest places of biodiversity anywhere, because we have all these very many species in this very small compact room. Yeah, so welcome. So I think what we'll do is we'll just kind of walk around the room, and we'll talk about things as we go, and then we'll do questions at the end. So first, I'd like to introduce you to Ms. Emily Saling. Emily is her 3rd day here as kind of an intern-like person. And so we're very excited to have her. Thanks, Emily. So when we first come in the door, we see that we have all these black boxes here. And these are desiccation chambers. So with except for one, that's a rehydration chamber. So what I mentioned earlier is that not all seeds behave the same way when you try to apply these seed conservation practices to them. And so because we're a small seed bank and because our flora is so threatened, it allows us to accommodate these species who behave in different ways when we try to store them. So we set the relative humidity inside of these boxes. We put the seeds inside and they're sealed. And then the seeds and the ambient air inside the chambers come into equilibrium with each other, or at least nearly so. We then hermetically seal them, and then we can cool them from there. And then the relative humidity inside of the packet also becomes into equilibrium with the seeds as well. We also, one of these is actually a rehydration chamber. That's because when seeds have been stored when they're dry, that if they come into contact with liquid water immediately, they can get imbibition damage, where they imbibe or drink in water so quickly that they get damaged. And so we rehydrate them for a short period of time first before we sow them and they come into contact with liquid water. Okay, so the microwave we mostly just use to sterilize agar, which we mostly use for fern spores and orchid seeds. And then we have two ovens down here. The oven on the left we use for determining moisture content of seeds. And then the one on the right is an old paraffin oven that I've retrofitted with a temperature control. So we're able to get a constant temperature incubation out of that. We can also use this to artificially age seeds or experimentally age seeds if we need to do that as well. Moving along, we have this hood. So unlike the hood in your chemistry lab, which is designed to vacuum toxic vapors out of the building, this works in reverse. So air comes to the top through a HEPA filter and then is blown out towards you in laminar flow. It's called a laminar flow hood. This we're really only using for fern spores and orchid seeds where we need fairly sterile conditions. And as you notice, you can see Emily over here kind of sowing seeds out in the open using unsterilized instruments, and that's all by design. If a seed gets parasitized by a fungus, and that's mostly what we see, that seed was probably never very healthy to begin with. So a lot of times what we'll see is maybe one or two seeds will turn into these little puff balls. And so you know that the surfaces of all the other seeds in that petri dish are loaded up with spores, but they're just fine. Yes. Okay. So the next thing that we have here are our -18 or -20 degree freezers. This is where the orthodox seeds are stored in here. Yes. Okay. Aha. The next thing I'll show you is this iron. I mean, I know I want to look good for you guys when I do a tour, but the other reason that we might have an iron is because of the storage media that we use for seeds. So if our number one criteria for storage media is being airtight or moisture tight, people have tested all kinds of different materials, including Ziploc bags, tin cans, every kind of plastic you can think of, silica glass, and trilaminate aluminum foil packs. And it turns out that these trilaminate aluminum foil packs perform about equally well to silica glass as far as being moisture proof. But these have several advantages. One, they exclude light, and so that's good. They also take up less space. They're more cost effective. I can drop them and they're not going to break, which is really nice. And also they're self-sealing. So if you were using glass somewhere, you would have a gasket or an O-ring, and then over time that would fail. That's what they're seeing in some other places. But these, because they're self-sealing, we don't have to worry about a gasket or an O-ring failing. So these are the kind of the storage media of choice because of that. And we seal them with an iron. Yes. Okay. We don't have access to DI water, but we make distilled water in batches. That's what this is here. Okay. So this is storage at +5. This is where we store some of those freeze-sensitive intermediate seeds and maybe some of the seeds that we might be sensitive to desiccation as well. Yes, okay. So then we have a -80 degree freezer. This is kind of our third storage temperature. The negative 80s are kind of a dime a dozen in molecular and medical labs, but they're not used so much in seed banking. But the reason why we have these might be because of these freeze sensitive seeds I talked about earlier, where there's that lipid phase state change at our conventional -20 storage temperature. But there's no lipid phase state changes, we think, happening pretty much past -40. So -80 might be a safer temperature to store these things. So that's why we have these. So one of the first to be used in seed banking. It's kind of fun to open. It's dramatic with the air, the cool air falling. Yes, we also store some ferns and some pollen in here as well. It's kind of fun. It knows some things like the ambient temperature and how many times the door has been opened and the last time the door has been opened. It has a name. It's Freezer von Koldelot. And it certainly is cool. Yeah. Okay. So moving on. This is one of our germination chambers. This is a machine that allows us to control some of the environmental variables with respect to the lighting and temperature regimes. So for example, this one is currently set to 25 degrees during the day and 15 degrees during the night. So for example, if I open it, we'll see that the lights are on. So it's in the day phase. And so it's 25 degrees. So it's warmer in there than it is in here. And yes, so we can see here's some germinants of a common bellflower family plant. This is Claremontia fowleri. Let's see. Aha. This is an endangered species. You can see that any green on there. Those are the cotyledons. In this lab, we define germination as radical emergence. So this is pretty far along, actually. Because we have a nursery at our same institution, and because our plants are so rare, what we do is when we have a germination event, we'll actually transplant that little seedling into a new petri dish, and then we'll send them down to the nursery for further propagation. Kind of giving these seeds or seedlings a second chance on life. In most seed labs, the seedlings are just discarded or incinerated in some way. So that's a great relationship that we have. Okay. Oh, so the balance I like to talk about. So the balance is what are the most kind of commonplace pieces of lab equipment anywhere. But measuring the mass of seeds is one of the only tests that we can do that's non-destructive. If we measure moisture content, that's non-destructive. Or if we measure the embryos, that's destructive. Even germination is technically destructive because we don't have those seeds in our collection anymore. And then we don't have any other questions to ask of them. But measuring the mass of seeds is non-destructive. So we can relate that back to destructive things like germination or environmental variables like average annual temperature or warmest temperature of the month or precipitation or what have you. Yes, and then we have two more smaller germination chambers over here in the corner as well. Yes, I could show you other cool things like how we print our petri dish labels and our seed pack labels straight out of the database so that there's no chance for any errors to be introduced. I should also tell you that in each one of these chambers, whether it's a desiccation chamber or a germination chamber, we have data loggers logging the temperature and humidity in real time on a cloud-based platform. So anywhere in the world, I can log in to the server and I can see, oh, what is the real temperature of the -18 storage, or what is the real relative humidity of this desiccation chamber? I also get an email and a text message if something gets too hot or too warm, or too wet or too dry. And so I can then check on these things in real time, which helps me sleep at night. Yes. Okay, and I think we're out of time. Yes? Perfect, great. So I'll see you back on my office. Aloha.
Thanks again, Dustin. So I saw some questions come in. Awesome. So we'll let him get back to his desk and then we'll get started on those. One that I can answer, somebody was asking about volunteers. We do have volunteers throughout our Botanical Research Center. In addition to helping out in our seed bank and laboratory, they also help out with the herbarium too. Hey, Dustin.
Hello.
So first question I have for you, Sherry was wondering, she says she's a fan of seed banks, and are they doing well in general? Is there a sufficient support in the seed world?
Well, yes. So there are actually a few thousand seed banks globally today. So back in the early 90s, that was not really a thing. Most of the seed banks or gene banks in the world were focused on agricultural species. They're kind of these crop gene banks. But today, and especially because of the role of botanical gardens in conservation, there are quite a few seed banks. One of the things that I didn't share is that we've put together a seed conservation directory of expertise. It's this interactive dashboard to facilitate knowledge sharing globally. So you kind of click on one thing and all the other things change. And so are they doing well? There's kind of different levels of seed banks. So I think a lot of these seed banks, they kind of don't do some of the curation type stuff that we talked about today. Like maybe they don't do the longevity testing. They do a lot of black boxing, which is still great. Yeah.
Thank you. And
we can always use more funding, right?
Yeah. I had another question come in from Jasmine. She's wondering, how long does the seed rehydration period usually take?
No more than 24 hours, because these are the exact kind of conditions where you can get mold to grow. And so we don't want that. Yeah, so 24 hours at the absolute most. So usually it's an overnight thing.
And Francine was curious about the generator. Does it support all the equipment in the seed lab in case of a power failure?
Yeah.
And she's also curious if it supports the rare book room and herbarium.
Yeah, so I should have mentioned that the seed lab is located in the Juliet Rice Wickman Botanical Research Center, which is a hurricane category 4 resistant building. There's also a backup generator that automatically kicks on in the event of a power loss. And so, yes, all of the seed bank, all of the storage equipment and the seed bank is supported by the generator. And I'm not sure about the rare book room. I imagine so. Yeah, but it's actually tested once a month. And so, conceivably, as long as there's diesel still on the island, then the seed bank will continue to run.
Thank you. And let's try to get maybe another quick one in there before you start your second part. Nancy was wondering, who and how can seeds be removed from the seed bank?
Yeah. So that's a good question. So we have a number of species managers, land managers, and lots of different people working with species or with places. So yeah, kind of anyone who's interested in a restoration project can withdraw seeds. A lot of times what happens is that the seeds are withdrawn and maybe we'll start their propagation up here, or maybe we'll send them straight to our conservation nursery, and they'll produce more mature plants than a seed and then outplant the plants. There's not so much direct seeding that happens kind of in general in Hawaii, but there's very low or no direct seeding that comes straight out of the seed bank. The vast majority of our collections are these rare and imperiled species of conservation importance. And so we're keeping maternal lines separate in a lot of these cases, which means that the number of seeds in most of our accessions are quite low.
Great, thank you. So let's go ahead and get started on your last little presentation you have for us. And if we didn't get to your question, we'll try to catch it at the end. So without further ado, Dustin, I'll let you take it away.
Okay. So now we're going to talk about a little bit about Ohia. So Ohia is kind of the most iconic plant in the Hawaiian flora. This picture was taken in Upper Limahuli Preserve. It is quite a beautiful plant. But it also has quite a bit of ecological and cultural significance. So, for example, it is one of the first plants that colonizes lava flows. And so it kind of creates substrate for other plants. So it's an early lava flow colonizer. It's the largest, makes up the largest portion of the canopy in our native wet and mesic forests. And so it provides habitat and nectar for native birds like apapane and also EEV. It also provides habitat for innumerable insects, snails, and other invertebrates. And it also acts as a nurse log. No, not that kind of a nurse log, although that is Ohia, but this kind of a nurse log where other plants will literally grow right on top of it. It also has a big role in replenishing our aquifers, kind of catching the mist out of our cloud forests. It's also incredibly culturally significant. So you see it in mo'olelo or stories, and mele or songs, uli or chants, and kininau or physical manifestations of deities. So for example, you see this in ku and kaane, laka, and pele, of course, the red flowers, and also hi'iaka, who's the only deity actually born in Hawaii. Another culturally significant is in construction. So you see it in construction of sacred places. For example, this is Pi'ilani Hale Heiau in the Kahanu Garden on Maui, which is one of the National Tropical Botanical Gardens. It's also a term used in battle for the kind of the first warrior to fall, has this term. You also see it in commerce. This is Ohia Hale. And you can see it is kind of makes beautiful flooring and beams here. You could also at one point buy it on Craigslist as firewood, which as we'll see in a moment is bad. And it's also important in the nursery trade as well. The Metrosideros, the genus, it's a pretty big genus, kind of found throughout the Pacific. There's about 60 or so species. But in Hawaii, all of the Hawaii native Metrosideros are actually endemic to Hawaii. And so we have these different taxa in Hawaii across the top. And in Kauai, we have these four, two of them of which are endemic to just Kauai. So they occur on Kauai Island and nowhere else in the world. And if you're on Kauai, they're actually very easy to distinguish from each other because you don't even need flowers. All you really need is a leaf. And by just the leaf characters, you can determine the taxa, which I have shown here. Yes, so the ancestral position is on Kauai about 3.9 million years ago. That was in the Pliocene, if that's interesting. And one of our very early foresters, this Griffith, he says that Ohia occurs far more frequently than any other species. And because of its influence on the ecosystem, where if you remove the species from the ecosystem collapses, it is therefore a foundational species, not to be confused with a keystone species. Yes. And so it's extremely important to the ecosystem and culture. And what our current forester, J.B. Friday, has this quote: "It's arguably the most vitally important species in Hawaiian forests." So it's, okay, extremely important. But enter rapid Ohia death, dun, 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 dun. So what has happened is that several years ago, residents of the Big Island started noticing that their trees, the leaves would turn brown, would rapidly defoliate, and then the whole tree would be dead in a matter of weeks. And so this phenomenon was colloquially termed rapid Ohia death. So that was from the air. This is kind of from the ground. What they're noticing now is that the rare species in the understory are also disappearing. Yes, so there's the brown leaves and then dead in a matter of weeks. That's the rapid part. Yeah. So it's now been detected on several of the islands. So this is Big Island and Kauai and Oahu, shown here, kind of where we detected it. And it's actually from two newly described species of Ceratocystis fungi. So these are species that are new to science, these only described in 2018. And Ceratocystis is a pretty big species complex. And so within this, they're not super closely related. There's actually two different species. So Ceratocystis leuco-Ohia, I've given it kind of a Hawaiian name. It means destroyer of Ohia, and that causes a wilt. That's the one where the whole tree dies. But there's also this Ceratocystis huli-Ohia, or changer of Ohia, and that forms a canker. And so only the downstream portion of those plants will die. This one is also harder to detect because you have to find where the tree is infected. Whereas in leuco-Ohia, you can sample kind of almost anywhere and find it more easily. So this is kind of what the stereotypical staining might look like in the heartwood. And so the whole state is kind of in crisis mode. There was a summit held at the Capitol a few years ago, and there are these rapid Ohia death strategic response plans. And in those, seed collection and banking has been identified as a critical strategy to kind of have a genetic safety net to capture the alleles of these plants. And so we got some funding from the Hawaii Tourism Authority, which is interesting for the state to recognize that these plants are important for the tourism economy. And so it kind of had three parts where we'd go out and we'd collect seeds, and we would bank these seeds and then reciprocate them with other seed banks. And then there was a community outreach aspect as well, which I won't be talking about. And so we went out and we collected seeds from Ohia. Then COVID hit and we had limited access to the lab, but we had all of these data. And so we thought, maybe we can see if there's a correlation with seed zone and environmental variables. So a seed zone is an area which you can remove seeds anywhere from that area and then put them back anywhere in that same area with a higher likelihood that those seeds will be adapted to that area. And so we made those with the states and others for Kauai. And then we looked at these 19 kind of classic bioclimatic variables that we calculated from a local source. And we found that there was no correlation with any of the environmental variables, but there was between two of the seed zones, with germination being lower in the Nepali valleys and higher in the windward ranges. And so the jury is still out on why that might be, but what this shows is that seed zones and also repurposed, routinely collected seed bank data can be important for conservation planning. So maybe you would, for example, collect more seeds in the Nepali valleys than you would in the other areas, for example. And so, so important for conservation planning. We also looked at another study where we harvested seeds of Ohia from the herbarium. So I told you earlier, like how that might be an overlooked repository for species or alleles or genes within a species. And so we came up with this really cool diagram to explain what happened to every single seed from every specimen over time, going back several decades from our herbarium. Not one seed from any specimen germinated. But we didn't give up. We did that other study with the pet plants that I shared earlier. We did, however, looked at freshly collected seeds and the oldest seeds that we had in the herbarium, in the seed bank at the time, which was 6.5 years old. And although not exactly statistically comparable, we found that the fresh seeds germinated at about the same as the oldest seeds, the 6.5 years. So the takeaway here is that seed banks are the winner. They're an essential tool in conservation science and practice. And I think we're there. Yes. And I will stop sharing this time.
Thanks again, Dustin. So I had, looks like a question from the previous presentation. Eric was wondering, are there specific methods for waking seeds from different types of dormancy?
Yes, absolutely. I have a whole lesson about this with activities, but so yes, there are. So if you can know something about what dormancy you have, then you can know something about how to wake them up. And yeah, there's, depending on which one of those you have, there's different methods. Yeah, and so that's something that we're really curious about and that we investigate here.
Great. So Sherry was wondering, is the fungus been there forever? If you replant, will the trees just succumb eventually?
Well, the fungus has not been here forever. So in the 1970s, they were noticing this phenomenon where large swaths of Ohia trees would kind of all die at once. And so they went looking for a pathogen. They went looking and they did not find a pathogen. And so what they were actually seeing is something called cohort senescence. So a whole stand of trees will all get established at the same time, kind of grow at the same time, and then they'll die at the same time. So that's how we know that they're new. Yes. So so as far as seeds go, no species of Ceratocystis, including the ones that cause rapid Ohia death, have ever been detected in seeds. So yeah.
Kim was wondering if you could clarify the distinction between foundational and keystone species.
Yes. So in both the foundational and the keystone species, if you remove that species from the ecosystem collapses. The difference is that in a keystone species, the biomass is disproportionate to the ecosystem services they provide. So because Ohia is kind of one of the biggest trees and the most abundant species, it cannot be a keystone species, but the ecosystem will still collapse if you remove it. So it's foundational.
Thanks. And she was also wondering if you are publishing your results of the seed zone data study.
Yeah, that's in review now.
And yes, Kim, we will be making the video available eventually on social media. Eric was wondering, he says, I know there was a goal of seed banking a certain % of Ohia. How are we doing on that goal?
Yeah, so we have some really lofty goals that has to do with collecting from a certain number of trees per taxon per seed zone in which they occur. And yeah, so it's quite a lot. So we're treating Ohia almost like we would treat rare species, where we collect from single individuals. So we're keeping maternal lines separate. So we collect from separate moms, and we'll go out and in most cases we'll tag the actual plant, because the best GPS in the world isn't going to differentiate between this tree and this tree, right? And so some of these seeds are also being used in resistance testing. Those are being sent to Hilo for some resistance testing. And so if resistance is found, they can then go back out into the field and go back to that exact plant to collect more seeds from one that might be resistant. So as far as how we're doing the statewide collecting goals, we got pretty far with that campaign. There were some other seed banks in the state that also got some funding to do collecting and banking. You can actually see these on Laukahi, I think in almost real time, or they're updated pretty frequently on the Laukahi website. Thanks, Nina.
And then Sherry asks, who mainly funds your work in the seed lab?
Well, okay, so everything I showed you except for one thing in the seed lab was bought on grants. So the tables that you saw, the back counter, the faucet, the Post-it notes, and the Sharpies, and everything in between, all the equipment was all bought on grants. The only thing that wasn't bought on grants is me. I am very thankfully on hard money, and everything else was bought on grants. So I am very happy to be supported institutionally by the National Tropical Botanical Garden. And so I think our budget before COVID used to break down to is about half earned income and about half donations and grants. Nina, do you want to chime in? I'm not sure after COVID. I don't know. Yeah.
Well, we can say it's many different types of funding that come. Some of it is indeed core funding. Some of it is from the membership that we have of the garden, from our trustees and fellows, but a lot of it also comes from science and conservation grants. So it could be the National Science Foundation, but also a lot from National Geographic Society or Mohammed bin Zayed and so on. So we'll go for any source that's going to support us. Thanks.
Yeah, that's right.
Well, thank you everybody for joining us today. We really appreciate you coming and attending live. So we will be sending out the recording to you shortly. We'll follow up in an email. And we'd also love it if you helped us improve our future sessions by taking about 5 minutes to fill out a short survey for us. That'll really help inform us what you thought about today and what we can do better next time. So thank you again. And if Dustin, Nina, you have anything else you'd like to say, go for it. They look good. Happy campers over there. Well, thank you again, everybody, for joining, and we'll hopefully see you at the next one. Ahui ho.